name is Chris Kochwar. My specific purpose is that I will persuade the class about the benefits of CRISPR gene editing. My central idea is that CRISPR gene editing is the pathway to a better future. Imagine a world where you could choose your child's hair color, eye color, and even their gender. Imagine the moment your parents could choose to eliminate any chance that you would have cancer. Imagine a place where vaccines wouldn't be necessary. Imagine making these choices for your own children. This perfect world is closer than we realize. Today, I will persuade the class about the benefits of CRISPR gene editing. Everyone has been affected by genetic disorders at some point in their lives, whether directly or indirectly. After conducting extensive research and reading many scientific journals, I have submerged myself in the knowledge of CRISPR's uses and benefits. We'll discuss what exactly CRISPR gene editing is, how it is being used today, and how it will benefit us in the future. First, allow me to introduce you to the fascinating technology known as CRISPR. So, CRISPR gene editing has many benefits, but to understand them, you must understand what the technology does. A group of scientists led by Brooks in 2014 wrote a paper which described how gene editing has been under development for around 12 years. CRISPR gene editing stands for clusters of regularly interspaced short palindromic repeats. So there are two parts to the CRISPR systems. The first is the Cas9 technology, which is an enzyme that can cut through DNA like a pair of nano-scissors. Second is a small RNA molecule that tells the Cas9 systems where to go to cut the DNA, and then the DNA mends itself. Further analysis was done by King in 2016, stating that CRISPR systems can make it possible to efficiently turn off or on genes in an organism's cells. It has been discovered that human embryos can be modified in a similar way. So here we have a DNA sequence. Each color is a different code for a different gene. So what the nano-scissors would do is they cut out, for instance, this red part and replace it with a similar red part, but they would change some of the symbols for it. So yeah. So now that you know what CRISPR is, you can learn how it is used today. CRISPR gene editing research and experiments is greatly benefiting our society today. During the early stages of CRISPR development, according to Brooks in 2014, tests were done on plants such as tomatoes because they have a really good quality of genome. The tests done on tomato plants ended up changing the phenotype, or the physical makeup of the plants, according to Brooks in 2014. This change allowed the plants to produce more fruits and grow bigger, which is a really good first step for this CRISPR systems. The first process that scientists say talked about in an article she wrote in 2019 is a process that is supposed to get rid of blood diseases, disorders, and cancer in patients. For these experiments, the researchers extracted some of the infected cells, they changed the DNA, and then put it back into the patient. One child that is coming up in the future is that people who inherit blindness will have the same process done to their eyes and will get their eye genes changed with the Cas9 nanoscissors. Say states that if these trials do yield excellent results, the cure for diseases like cystic fibrosis and thoracic cancer are in arm's reach. A research group led by King in 2016 explained how CRISPR has its limitations, but it may be used to treat HIV. Currently, HIV has no cure, but if CRISPR can more accurately delete genes, there will be a cure for HIV. To test this technology, studies on human embryos and clinical trials would be required. Now that you understand what CRISPR is and how it is used today, we can discover the possibilities for the future. CRISPR gene editing will yield a multitude of benefits for future generations. One of the first concerns with CRISPR is that there will have to be many laws placed on the technology and who can use it, according to Detweiler in 2018. A scientist from China claimed to have used CRISPR on 3 human embryos. 2 of these embryos were put back into 2 different women, and they gave birth to twin girls. Now, this scientist later got arrested for improper use of medical equipment, but they gave birth to 2 different twin girls. So any laws that would have to be put on CRISPR would have to be passed by departments such as the U.S. Food, drug administrations, which would be a really long process. A research paper written by Rosan in 2018 states that uprising and debates have been vigorously discussed over the many years about the testing on live human embryos. People find this unethical, but for some tests, like the one I just talked about, they are trying to put it back into the female so that they can give birth. CRISPR can be used to cure diseases, which means that it can affect future generations in a big way. Dr. Nada published a book in 2014 about vaccines and the long-lasting effects they can have. Vaccines are used to cure diseases, but some people are allergic to the vaccine or their bodies react negatively to the medicine. Trials for vaccines are long-lasting, dangerous, and costly, but with CRISPR, we can cut the cost in half and double the amount of lives saved. So CRISPR will have long-lasting, life-changing effects. These are some examples of diseases that CRISPR can cure. They include cancer, blood diseases, blindness, AIDS, cystic fibrosis, muscular dystrophy, and Huntington's disease. So today, I persuaded the class about the benefits of CRISPR gene editing. After conducting research and reading scientific journals, I was able to persuade you about the benefits of CRISPR gene editing. First, we discussed what exactly CRISPR gene editing is and the different parts to it. Then, we dove through the uses of CRISPR today. Finally, we slid through the uses that CRISPR will eventually have in today's society. Everyone knows someone who has cancer or a genetic disorder, which means everyone would be affected by this technology if it becomes legalized and more legitimized. Imagine a perfect world where you wouldn't have to lose anyone to disease or cancer ever again. If you could save the lives of your friends, would you?